Welcome back to this next video in which we are studying headaches. Okay, so in this next video we're now going to discuss secondary headaches, which remember are headaches that are unpleasant and the underlying pathophysiology is dangerous. So, topics that we're going to go over intracranial hypertension first. We will then go over low pressure headaches, which are intracranial hypotension. We'll then go over infectious causes of headaches, such as meningitis and sinusitis. We'll go over trigeminal neuralgia, which is a form of neuropathic pain. We'll then go over giant cell arteritis, and we'll end the video by going over glaucoma. Okay, so let's begin then with intracranial hypertension. So what does this mean firstly? Intracranial obviously means inside the skull. Hypertension means too high pressure inside the skull. So intracranial hypertension is too high pressure inside of the skull, and we often abbreviate it down to ICH for short. Now, another little abbreviation that we often use ICP to denote intracranial pressure. And obviously intracranial hypertension, you could also refer to that as raised intracranial pressure. The pressure inside the skull is going to be raised. Now, we will come back later to discuss what sort of things can lead to the intracranial pressure being raised. There are all sorts of different things that can lead to raised intracranial pressure. Probably the most feared and most famous is, of course, a brain tumour, but there are loads of other things that can lead to raised intracranial pressure besides brain tumours. However, if you want a little example to be thinking of, a brain tumour would create raised intracranial pressure. Okay, so what I now want to discuss is a description of the actual headache that you're going to get in intracranial hypertension, because it doesn't matter what the cause of the raised intracranial pressure is. It doesn't matter whether it's a brain tumour or whether it's something else that is causing the raised intracranial pressure. The actual headache that you're going to get as a result of having too high intracranial pressure is going to be the same, no matter what the different causes are. So let's begin by describing this. So, if you have raised intracranial pressure, why is that going to give rise to a headache? Well, it's because the meningeal nociceptors are going to get pushed on. When you have raised intracranial pressure, that means that everything is going to be trying to bulge out of the skull more than previously, and therefore the pressure on the meningeal nociceptors is going to be increased. And when the pressure on meningeal nociceptors is increased, that is going to lead to them activating. So raised intracranial pressure is going to be activating meningeal nociceptors. So we'll draw a little flow diagram here. Raised intracranial pressure is going to be activating the meningeal nociceptors, and it's going to be activating the meningeal nociceptors everywhere in the meninges. So this is something interesting. When we were discussing migraine headaches, which also resulted in the activation of the meningeal nociceptors, it was just the meningeal nociceptors in the region of the meninges over the area that has actually undergone the cortical spreading depression that were going to actually be activated. Whereas if you've got raised intracranial pressure, that's everywhere in the cranial cavity, so you'll be activating meningeal nociceptors everywhere. So, at this point, it's probably going to be helpful for me to have a picture of the brain again. So I'm just going to draw one of my quick pictures of the brain where we view it from the left-hand side. So I'll just do this here. So there, of course, is the cerebral hemispheres viewed from the left-hand side, left cerebral hemisphere specifically, the bottom portion of the brainstem there, the pons, and then the medulla, and then, of course, we have the cerebellum here. Okay, so here's my basic picture of the brain, and of course it will be sat within the cranial cavity and it will be covered by the meninges. Now, let's try and understand where the site of the headache is going to be in intracranial hypertension. So you're going to be getting activation of the meningeal nociceptors all over the brain here. On both sides is the first thing, which means that this headache is going to be bilateral. So let's start by writing down some of the descriptions here. So the headache that you're going to get in intracranial hypertension is going to be bilateral. It's going to be on both sides because the meninges on both the left-hand side and on the right-hand side, the nociceptors there are going to be activated. Now, thinking more in detail about the sites that are going to actually be painful. We've talked about where activation of the meningeal nociceptors, where that pain actually refers to. We discussed that at the front of the brain, the frontal region and the orbitofrontal region, and also all the way along the top of the brain, and then the occipital region of the brain, all of these meninges, they are innervated by primary nociceptive afferents that will come in the ophthalmic nerve and will synapse onto the same second order neurons as primary nociceptive afferents that have run in the ophthalmic nerve from peripheral sites. And therefore, when you activate meningeal nociceptors in this sort of domain, and I might even actually just draw a little bit of it on, so this bit that I'm marking out in dashed red lines, all of this region here, when you activate the meninges over that region, we know that that refers to the frontal region of the head on the same side, of course. So on the left, this will refer to the left frontal region and also the periorbital region potentially as well, which is also innervated by the ophthalmic nerve. So into the ophthalmic areas. Okay, then we discussed that this lateral portion here, which is going to be innervated by branches from the maxillary nerve at the front and from the mandibular nerve at the back, this refers to the temporal region. So the anterior portion is very anterior temporal region, and the mandibular region back here is more posterior temporal region, that region of the face just lateral to the orbits, to the eyes. Okay, so we'll have it also going into the temporal region if we activate those portions of meninges there. And of course we know that with regards to the meninges back here, covering the cerebellum and covering the lower portions of the brainstem, the portions of meninges in the inferior posterior cranial fossa, they refer, we know, to the back of the head region, the occipital region. And the reason is that they are innervated by primary nociceptive afferents that come in the posterior rami of the first, second, and third cervical mixed spinal nerves. And of course, they also bring in peripheral primary nociceptive afferents from the back of the head and the back of the neck region. So this will refer to the occipital region. And I should have tried to colour code this better. So temporal region, coloured in green here, this is all of this portion of meninges here, and then the bottom of the brain here, the cerebellum and the bottom of the brainstem, that is the occipital region of the head. So why have I just gone over this? It's to emphasise to you that it's going to be all over, because we're going to be activating the meninges all over. Therefore, you're going to get a headache in the frontal region, potentially the periorbital region, the temporal region, and the occipital region, and it's going to be bilateral. So you're going to get this headache that is going to be experienced all over the place. Okay, so that's the site. With regards to severity, of course, the severity will depend on how many meningeal nociceptors you're going to activate, and also how frequently the meningeal nociceptive afferents will be firing. And how frequently the meningeal nociceptive afferents will be firing will depend on how big the intracranial pressure rise is. So if you have a massive intracranial pressure, a massive intracranial hypertension, that will cause a bigger, worse, more severe headache all over than if it's only just raised. So the more raised the intracranial pressure is, the worse will be the severity. So severity can vary depending on how big the rise in intracranial pressure is. So, we've done site, we've done severity, now we should discuss character. We might be able to guess what the character of this headache is going to be. Think, we are activating the meningeal nociceptors. We know that the meningeal nociceptors, most of them are surrounding meningeal arteries or extraaxial brain arteries. And therefore, if they're activated, their activation is going to be increased when the arteries pulse. So when systolic blood pressure is present and all the arteries expand, that will increase the activation of those meningeal nociceptors if they're already activated. And therefore the primary nociceptive afferents will be firing at a greater frequency during systole than during diastole. Therefore, the pain will pulsate in tune with the pulse. So I'll put this here. So the pain is pulsating in character. Okay, so I've now given you a basic description of the pain that you're going to have in intracranial hypertension, and I hope you understand where I've come up with all of these descriptions from. It's going to be bilateral because the meningeal nociceptors everywhere are going to be activated. It's going to be in these regions bilaterally because that's where the pain from the meninges refers to, so it's going to effectively be all over the head. And it's going to be pulsating in nature because of the fact that the meningeal nociceptors surround arteries mainly, and therefore this is the same reason that migraine headaches are usually pulsating. Now we should talk about when is the headache actually present. Well, the headache is most likely going to be present every single day, and it might be the case that it's present all throughout the day, i.e. that permanently you have this headache. However, it's not necessarily present throughout the entire day, because generally what happens with intracranial hypertension, and this is in fact what we'll spend the rest of the video trying to understand, what happens in intracranial hypertension is that the headache is generally worse when you just get up. In the morning, intracranial hypertension is worse, the headache is much, much worse, and then it gets better as the day goes on. And sometimes it can actually go away completely in the afternoon. In other people who have more severe intracranial hypertension, it might be the case that it just gets better in the afternoon, but it's still present, and therefore it's present all day, it's just worse in the morning. But in some people with more mild intracranial hypertension, it's bad in the morning, but then it can get completely better in the afternoon and go. But it will be there every day until the intracranial pressure is dealt with and brought down, until whatever it is that's causing the raised intracranial pressure has gone. Okay, so it's present every day, but it's not necessarily present every hour of every day. It might be the case that it's worse in the morning, but then it's gone in the afternoon, or it might be the case that it is present all the time, it's just worse in the morning. But this is a very important feature of intracranial hypertension's headache, that it is worse in the morning. And I now want to spend a long time discussing with you exactly why it is worse in the morning and then better in the afternoon. Okay, so let's begin this story. Why is the headache going to be worse in the morning and better in the afternoon? Well, let's think about this logically. The headache is being caused by the raised intracranial pressure activating the meningeal nociceptors. If the pain goes away in the afternoon, what does that mean? That means that there is less activation of the meningeal nociceptors, which is producing then a less frequent firing of the primary nociceptive afferents. If there is less activation of the meningeal nociceptors, that must mean that the intracranial pressure is lower in the afternoon. And that's exactly what happens. Intracranial pressure, even in healthy individuals, obeys a circadian cycle. It goes up and down on a 24-hour rhythm, and in normal people it is usually highest in the morning and it goes down in the afternoon. When you have intracranial hypertension, therefore, it's going to be worse in the morning because as the normal part of the circadian fluctuation, intracranial pressure is higher in the morning, and then in the afternoon it might now go down into a level that is not actually capable of activating the meningeal nociceptors. So I think what would be helpful is drawing another circadian graph. So here we go. So we've drawn one of these previously when we were discussing the activation of the suprachiasmatic nuclei. So let's put time along the x-axis here, so going up in every 6 hours. So 0 a.m., 6 a.m., midday, 6 o'clock in the evening, and then midnight again. Okay, and this time on the y-axis we're going to put intracranial pressure, ICP here, and we want to see how it goes up and down. So, what's going to be happening is during the night, intracranial pressure is going to go up and up and up, and then by the time you get up in the morning, it's at its highest, and then as the day goes on, it will fall back down like so, and then as the night comes on, it will then go back up. So this is the sort of sinusoidal graph that we have for intracranial pressure. It's highest in the morning and then lowest in the evening. And this occurs, as I say, in everyone. This is a perfectly normal phenomenon that intracranial pressure varies on a circadian basis. It obeys a circadian rhythm. Now, if you have intracranial hypertension, therefore, so let's now draw this. In fact, I won't draw it in green. I was about to pick green as the colour. Let's draw it in red, evil red. Then your graph might now look like this. So the whole thing, you can see I've translated it upwards. So of course, the intracranial pressure is now going to be highest at the morning. And let's say, to activate the meningeal nociceptors, it has to be above this line, this dashed line that I've shown you here. So here is our normal person with normal intracranial pressure, and you see their intracranial pressure is never going to get above that line, therefore it's never going to activate meningeal nociceptors enough to cause pain. However, this person with intracranial hypertension here, you can see that their graph does go above this line, and it goes above this line in the morning, therefore they're going to get the bilateral pulsating headache all over the head in the morning, and then it's going to go away in the afternoon. Now, if this was extreme enough, you can imagine if I brought it right up here, then all day long it would be above this line, and therefore you would have a headache all day long. It would just be worse in the morning and better in the afternoon. But if it's not too severe, the intracranial hypertension, as I've shown here, then in the afternoon your graph will go below this line, and therefore you won't be getting pain anymore. You won't be getting the activation of the meningeal nociceptors. Okay, so isn't this lovely to know? Intracranial pressure fluctuates on a 24-hour basis. The question, of course, that you're probably shouting at your computer screens now, if you've got any curiosity at all, is why? Why does the pressure inside your head fluctuate on a 24-hour basis like this? And the answer is that it is something to do with the cerebrospinal fluid. So, I'm going to introduce you to a very nice little principle that is helpful for when thinking about intracranial hypertension. Indeed, will be very helpful when we come on to discuss different causes of intracranial hypertension. And this is quite pretentious. It's a very simple principle. However, it's given a great big pretentious name. It's known as the Monro-Kelly, and it's not even called the Monro-Kelly principle, it's called the Monro-Kelly doctrine. Okay, so it's got a very, very pretentious name, this very simple concept. And the Monro-Kelly doctrine, then, this simple concept, is that intracranial pressure depends on three things. There are three things inside the skull, and if you increase any one of them, that will increase intracranial pressure. That's what the Monro-Kelly doctrine is. So what are those three things inside the skull? Well, hopefully you can work this out. One of them, of course, is the brain. The brain is inside the skull. And the brain includes the meninges, by the way. We count all of the brain tissue, including the meninges, as tissue that's inside the intracranial fossa. Okay, or the intracranial cavity. So brain is one of the components inside of the skull. Another component is obviously cerebrospinal fluid, CSF, and then the final component is, of course, blood in the blood vessels. So, three things are inside the skull: brain, CSF, and blood, and if you have a raised intracranial pressure, that is because one of these has increased, or more of them have increased than one. Okay, so to change intracranial pressure, you have to change the amounts of these three things. That is what the Monro-Kelly doctrine is. It's a really simple concept, given an incredibly pretentious name, but people expect you to know that this principle is called the Monro-Kelly doctrine, so know that. So, if intracranial pressure is going to fluctuate on a 24-hour basis, that means that one of these, the volume of it, must be changing on the 24-hour basis. Is the volume of brain likely to be changing on the 24-hour basis? No, of course not. Is the amount of blood in the cranial cavity likely to be changing on a 24-hour basis? Well, it's more plausible that that might be changing, but really, on a 24-hour basis like this, do you expect the blood supply to be changing? You might argue that, when you're asleep, maybe the brain doesn't need as much blood because you're not using it as much. The answer is not really. The brain's blood supply pretty much stays the same. So, what we are left with then is that the amount of CSF is going to change on a 24-hour basis. Indeed, it's going to go up at night and it's going to fall during the day. So CSF in the cranial cavity is going to go up at night, and we need to try and understand why it's going to go up at night. So, for this, I now want to do my full discussion of cerebrospinal fluid. I want to discuss the ventricles inside the brain, and then I want to discuss how the ventricles are connected to the subarachnoid space. I want to discuss how CSF is produced within the ventricles, and then how the CSF will make its way into the subarachnoid space, and how it's going to be resorbed into dural venous sinuses through things called arachnoid granulations, majorly through the superior sagittal dural venous sinus. Okay, and then we can answer why CSF in the skull goes up during night and then comes down during day, and therefore we can fully understand why intracranial hypertension headache is going to be worse in the morning. Okay, so we're now going to do a bit of the anatomy of the ventricles inside the brain. So we've seen a lot of these already. So we want to discuss firstly the four ventricles, the first, second, third and fourth ventricles. So, we've seen the third ventricle and we've seen the fourth ventricle previously. We'll start by drawing those on, and then we'll put lateral ventricles on, which are the first and the second ventricles, which we haven't seen hugely before. So I'm going to draw them in as simple shapes as possible. So I'm going to draw the third ventricle here in blue, in a nice simple shape here, like so. So it's roughly a rectangular shape here, and of course it's surrounded by the thalami, so on either side of it you will have the thalami. I'm not drawing those on here. And then at the front it's got the hypothalamus making up its walls, and indeed its bottom. And at the back its bottom is made up by, of course, the midbrain that's sitting underneath it. So here is the third ventricle, and let's just label it up. So here, this is the third ventricle. Let's now connect the third ventricle to the fourth ventricle. So of course, that's via the cerebral aqueduct that passes through the midbrain, like so. So here is the cerebral aqueduct. So I'll just put a few labels on here. So this is the cerebral aqueduct that we have seen previously in all of our cross sections of the midbrain. So that's going through the midbrain, and then that's going to open up to the fourth ventricle, which, remember, is behind the pons. And the boundaries of the fourth ventricle are in front. The front wall is, of course, the back of the pons. The lateral walls are the cerebellar peduncles, the superior cerebellar peduncles, the middle cerebellar peduncles, and then the inferior cerebellar peduncles, and the posterior wall is the cerebellum behind. So let me now try and carefully show the sort of shape of the fourth ventricle. So viewed from the side like this, and I hope you gathered that we were viewing this from the side. I don't know if I explicitly told you that, but viewed from the side here, the fourth ventricle has this sort of triangular shape like so. So it goes backwards somewhat. It's quite a deep ventricle here. The cerebellum will be in this position, and of course the lateral walls will be the middle cerebellar peduncles at this sort of level, the superior cerebellar peduncles at this sort of level, and the inferior cerebellar peduncles at this sort of level. So, viewed from the side, that is the fourth ventricle here, and then of course we have below the central canal that runs through the medulla and then through the spinal cord. Okay, so this is some of the CSF inside of the brain, and this is something really important to understand. There are two locations for CSF inside the skull. There is CSF in the subarachnoid space that is around the brain, outside the pia mater, and there is CSF that is inside of the brain in the ventricle system and the cerebral aqueduct in the central canal that goes all the way down the spinal cord. We will discuss the communication between this intra-brain CSF and the subarachnoid CSF, and that's one of the crucial things that we need to discuss. And before we do that, however, of course, the gaping hole in this picture so far is the lateral ventricles are not shown on here. Now, I'm only going to show one of these. I'm going to show the left lateral ventricle. The right lateral ventricle would be the mirror image on the right-hand side. So, let's now mark it on, and I will do it in a different colour so that it shows up. So we'll do it in a much lighter blue colour here. So this is roughly, and I've just realised that that comment of that label of cerebral aqueduct is actually in the way of what I want to, where I want to draw the lateral ventricle. So it's going to go, so there goes the annotation, cerebral aqueduct, and now let's draw on the lateral ventricle, the left lateral ventricle. Okay, so here we go. So it is above the third ventricle in this sort of position, like so. And we've seen it previously in that coronal section that we drew of the brain, where we were trying to understand where the middle cerebral artery is intraaxial for a part. Okay, so this is roughly the shape of the lateral ventricle, and of course I said when we were taking that coronal section that we could not see the lateral ventricle's shape properly because all we could see was a cut surface of the lateral ventricle. We had effectively taken a cross-section down here and we could see the lateral ventricles on either side. We could see just this little bit here. We could not see the whole thing going back like this. So this is the shape, roughly, of the lateral ventricle then, on the left-hand side, and imagine the mirror image on the right-hand side. So different parts, so we split it up into different parts like so. We split it into these 4 different parts. So this bit in the middle, I'll start here. This is called the body of the lateral ventricle. This bit at the front, which extends into the frontal lobe of the cerebral hemisphere, in this case the left cerebral hemisphere, this is called the anterior horn of the lateral ventricle. This bit right at the back that will extend into the occipital lobe of the left cerebral hemisphere, that's called the posterior horn, and this bit at the bottom, which extends into the temporal lobe of the left cerebral hemisphere, this is called the inferior horn. Okay, so this is the shape of the left lateral ventricle, and again this is full of CSF. So this is a great big hollow structure in the left cerebral hemisphere. There's the mirror image in the right cerebral hemisphere, and it's full of CSF, and it communicates with the third ventricle. So there is a connection between the lateral ventricle and the third ventricle, and I'll just put that in here. Okay, so this is known as the interventricular foramen, and of course you'll have 2 interventricular foramina because you'll have one into each of the lateral ventricles. This is the one into the left lateral ventricle. There will be another one on the right hand side which will connect the third ventricle with the right lateral ventricle. So this is the interventricular foramen, and it's also called the foramen of Monro, or if you want to give it its full name, you can call it the interventricular foramen of Monro. So this is the left foramen of Monro. There will be a right foramen of Monro going into the right lateral ventricle. And I don't know if I've said this previously in earlier videos, but the 2 lateral ventricles, they are the first and second ventricles, but you don't decide which one to call the first one and which one to call the second one. It isn't the case that someone has decided that we'll call the left one the first one and the right one the second one. So there's no consensus on which one should be called number 1 and which one should be called number 2, but together they are number 1 and number 2, but they're the lateral ventricles. Okay, so this is the way that the lateral ventricles are shaped, and this is the way that they connect with the third ventricle. So we now have seen the complete ventricular system that is full of CSF and is inside of the brain. Now, the ventricular system is actually where the CSF is going to be created. The lining of the ventricular system is called ependyma. So I'll write this down. So the epithelium that lines the ventricles, that is between the CSF inside and the brain material outside, this is called the ependyma. And there are special portions of the ependyma in the ventricles, in the lateral ventricles, in the third ventricle, and in the fourth ventricle that are creating CSF all the time. And these are called the choroid plexuses. So we refer to the type of ependyma which produces CSF as choroid plexus, and all over in loads of different sites in the lateral ventricles, in the third ventricle and the fourth ventricle, there will be sites where there are choroid plexus. So not all of the ependyma that lines these ventricles will be choroid plexus, but there is lots of choroid plexus in the ependyma of the 4 ventricles, and this is creating CSF. So, the ventricle system then, inside the brain, this is where CSF is being created. Now, we can't just continue on creating loads and loads of CSF and then not taking it away. And by the way, you might be wondering, well, what is the purpose of the CSF? Well, one of the purposes of the CSF is, of course, to cushion the brain. When it's in the subarachnoid space, it cushions the brain. But another purpose of it is to take waste products away from the brain material. Waste products can diffuse across the pia mater into the cerebrospinal fluid and indeed in the subarachnoid space, and indeed they can also diffuse across the ependyma into the CSF in the ventricles. So waste products from the brain can go into the CSF. Now, that means that we can need to continuously be replacing the CSF, otherwise it would just become full of waste products. So we're continuously producing more CSF from the choroid plexus sites in the ventricles. The question is, where does the CSF get resorbed into the venous blood? It gets resorbed in the subarachnoid space, specifically the subarachnoid space near the dural venous sinuses, particularly the superior sagittal sinus. And that, of course, is CSF in subarachnoid space. So we need somehow the CSF that is in the ventricles to be able to get into the subarachnoid space. So where is the communication between the ventricular system and the subarachnoid space? Well, this is at the fourth ventricle. So at the fourth ventricle, there are sites for communication with the subarachnoid space. So where are these sites? For this, it is helpful for me to draw a picture that I drew long ago, a picture of what the brainstem looks like from the back. And when we draw that picture, of course, we remove the cerebellum and we can see the boundaries of the fourth ventricle quite well. And this is extremely helpful for understanding where these foramina that connect it with the subarachnoid space is. Remember, of course, that the fourth ventricle is inside of brain, and then the brain is covered by pia mater. So to get out of the ventricular system and into the subarachnoid space, we need tubes that will pierce through the ependyma, through the brain, and through the pia mater, and then we can connect the CSF in the ventricle. The fourth ventricle is the one we're going to do these connections in to the subarachnoid space. So I'm now going to show you where these tubes exist. So let me draw this picture, and we'll do it in yellow here. So of course, we start with the midbrain up here, and when you draw a picture of the midbrain from behind, you see the colliculi. So here are the superior colliculi, and here are the inferior colliculi. Then we're going to see the pons below, and of course, we're going to see the superior cerebellar peduncles of the pons here, which connect, again, the pons with the cerebellum. The middle cerebellar peduncles, the big ones in the middle here, like so. And then at the base, of course, we have the inferior cerebellar peduncles, which extend into the medulla. They're not just at the back of the pons, they're also at the back of the medulla, like so. And then below, of course, we'll then have the medulla here. And on the back of the medulla, what will you be able to see? Some tests here. The dorsal column nuclei projecting out from the back here. And so, of course, these are the gracile nuclei, and these are the cuneate nuclei. Okay, so here then, this is the back of the pons, and in this space, this is where the fourth ventricle is. The cerebroaqueduct will run through the midbrain and empty in here. The central canal will then be the continuation down through the middle of the medulla there. Now, all of this surface is, of course, lined by pia mater, and then on top of that pia mater, you'll have subarachnoid space. So, we can make connections between the fourth ventricle and the subarachnoid space. The first two that I'm going to show you are the foramina of Luschka, and these are going to penetrate through at the junction, roughly, between the inferior cerebellar peduncle and the middle cerebellar peduncle. So in these spaces here, there is actually going to be a tube that connects the fourth ventricle to the subarachnoid space. So I'll just mark this in here. So there's a little tube there, and there's a little tube there. So understand exactly what's happening. You'll have a hole in the ependyma. You'll have a tunnel through the brain material here. You'll then have a hole in the pia mater. Of course, the whole tunnel will be lined by a continuum epithelium joining the ependyma to the pia mater. And then you've got CSF that can move from the fourth ventricle into the subarachnoid space around the brainstem here. And that's on both sides. You have two of these foramina, and these are called the foramina of Luschka, which is spelt, I believe, like so, Luschka. Okay, so you have the left foramen of Luschka and the right foramen of Luschka. Now there's another hole that we need to discuss that connects the fourth ventricle to the subarachnoid space, which is called the foramen of Magendie, and there's only one of these. So the foramen of Magendie is the next one we need to discuss, and this is right in the midline, and this is actually going to go into the cisterna magna. Now, do you remember me telling you about the cisterna magna? Come back up to this picture. Remember, the pia mater wraps perfectly around the brain, so it's wrapped into this little crevice between the cerebellum and the medulla, the back of the medulla there. The arachnoid mater and the other outer meninges don't wrap around the brain as well, and they come down like this to create a great big cistern, a great big collection of cerebrospinal fluid in the subarachnoid space there. And remember, that was called the cisterna magna, and I'll just put that in here. So that was called the cisterna magna. Now, that is in this position here on the back of the medulla. All of this is coated by pia mater, and on top of that is the subarachnoid space, and it's in this position. So there is going to be a connection between the back of the fourth ventricle here and that cisterna magna, and it's going to come in between the bottom of the two cerebellar peduncles here, the inferior cerebellar peduncles. So in this space here, again, what we're going to have is a tunnel through the ependyma, through the brain tissue, through the pia mater. That tube will be lined by an epithelium that is the continuation of the ependyma and the pia mater, and then CSF can come from the fourth ventricle to the cisterna magna, which is just part of the subarachnoid space, and that's called the foramen of Magendie. So, these are the connections then, the foramina of Luschka and the foramen of Magendie. These are the connections between the ventricular system CSF and the subarachnoid space CSF. So CSF is produced in the ventricles by the choroid plexus. It can then come out of these foramina into the subarachnoid space, and then in the subarachnoid space it's going to be resorbed at the dural venous sinuses, particularly at the superior sagittal sinus, and that's the one that I'll use as an example. So I want to show structures called the arachnoid granulations, which I have not previously shown. So, another picture then. So, what am I drawing here? This is meant to represent the skull, here in orange. So this is the top of the skull. I'm drawing a section of the superior sagittal sinus. So imagine this is the top of the skull right in the middle. So we can have the left cerebral hemisphere here and the right cerebral hemisphere here. We're looking in between. Okay, right. Then of course you will have the periosteal dura mater, which I'll put here in white. Okay, so all of that is periosteal dura mater. Then underneath the periosteal dura mater, you will have the meningeal dura mater, and this is the one that folds inwards to create the falx cerebri, and you'll also have, of course, the superior sagittal sinus in this position, like so here. Okay? So like that, and of course this is made up of two layers of meningeal dura mater, so I'll colour this in blue, in green, like so. So that's the meningeal dura mater, and I've made a mistake there, and the astute ones among you might recognise the mistake that I've made. There should not be that bit there, okay? The superior sagittal sinus is in between the meningeal dura mater and the periosteal dura mater, like so. So this is going to contain blood in there. Right. And then, of course, there will be the arachnoid mater around the outside. So I'll just put this on like so. So this is the arachnoid mater. Okay, so, and then, of course, underneath the arachnoid mater, you'll have the subarachnoid space. So why am I drawing this out? Of course, this is nice revision, but I now want to add something on that we have not previously discussed, which is something called an arachnoid granulation. And this is a way that the CSF in the subarachnoid space here, and I'll just label this up, this of course is the subarachnoid space here. This is a way for this CSF to be resorbed into the superior sagittal sinus here, this dural venous sinus. And these structures called arachnoid granulations are going to be all the way along the superior sagittal sinus. And they're also going to be present on other dural venous sinuses, although the superior sagittal sinus is the classical major example where there are loads of arachnoid granulations. So what is an arachnoid granulation? Well, it's actually quite difficult to show on this picture because I've done everything so thickly. But imagine making a hole in the meningeal dura mater that covers the superior sagittal sinus here, this dural venous sinus, and then putting a projection of the arachnoid mater inwards. So let me just draw another little picture here, a thinner version of this. So here is the meningeal dura mater here, like so, and there what you can see is I've made a little hole in the meningeal dura mater. So this is where the superior sagittal sinus here is, and now we're going to push the arachnoid mater through that little hole. So here in blue, here is the arachnoid mater, and now look, I can make this little extension of the arachnoid mater like that into the dural venous sinus, like so, and in this case, of course, this is the superior sagittal sinus. So I can make a projection of the arachnoid mater through the meningeal dura mater into that dural venous sinus, and I'll just colour this in blue so that it's clear what this is. And this is the concept of an arachnoid granulation, and CSF in the subarachnoid space can come into the lumen in the middle of this arachnoid granulation and then can cross the arachnoid mater into the venous blood in the dural venous sinus, and that is one way that CSF can be resorbed into the bloodstream, and that is the major way how CSF is resorbed into the bloodstream, through these arachnoid granulations in the dural venous sinuses. Okay, so that completes up my CSF story. We've discussed the ventricular system here. CSF is created in the ventricles by the choroid plexus. It then moves into the fourth ventricle through the foramina of Luschka and the foramen of Magendie into the subarachnoid space. It then moves around the subarachnoid space to where there are arachnoid granulations, and the CSF is then resorbed into the venous blood through the arachnoid granulations. Okay, so that's the control of CSF. So now we still have the question to answer: why is there going to be more CSF being produced or not being resorbed in the night, and then why does it go down in the day? Because I've said that this fluctuation, this circadian fluctuation in intracranial pressure, is caused by a circadian fluctuation in the amount of CSF. So why is the amount of CSF so much higher in the morning and then it goes down during the day? Well, the answer is that the amount of CSF that is being resorbed into the venous blood through the arachnoid granulations for some reason is lower during the night. So in the night here, your resorption of CSF into the venous blood goes down, and you're continuing to produce CSF by the choroid plexus. So that means now that you're going to build up CSF, and therefore the CSF gradually builds up through the night because you've got low resorption and production is still going on. And then once the daytime comes, then the resorption of CSF goes back up, and therefore the amount of CSF starts to fall down, and therefore the intracranial pressure falls back down. So this is all triggered by a circadian fluctuation in the resorption of CSF. So I'll just add the answer here. So it's worse in the evening because there is a circadian fluctuation in the resorption. It's worse in the morning rather. It's worse in the morning because overnight there is a circadian fluctuation in the resorption of CSF, and it is much, much lower in the evening. Whereas in the daytime the resorption of CSF goes up hugely. So, in the evening, when you're not resorbing much CSF, you're still producing more CSF from the choroid plexus, therefore the amount of CSF goes up and up and up, and this causes intracranial pressure to go up and up and up. So by morning, intracranial pressure is at its maximum, then resorption of CSF goes back up, so CSF is going to go down, and therefore intracranial pressure is going to fall back down, and then by the evening it's at its lowest, and then of course resorption of CSF goes back down, and then the whole thing repeats itself again. So this is the reason that intracranial pressure fluctuates on a 24-hour basis, and that's the reason that the headache is worse in the morning and might even go away in the afternoon. If it doesn't go away in the afternoon, it will at least be less severe in the afternoon. Okay, just one final thing that I want to say in this video before we have a break, and in the next video we'll go on to discuss accompanying symptoms of intracranial hypertension. The final thing that I'd like to add in this video is that it's also a classical headache that is worse when you strain. So it's worse when you sneeze, so worse when sneezing, and also worse when you cough. So worse when sneezing and coughing. Now, why is that? Well, it's because when you sneeze or cough, what happens? In the trunk of your body, the pressure inside the trunk of your body goes up when you sneeze and cough because you're trying to expel air out of your lungs, and to do that you force the pressure in your trunk, the major portion of your body, particularly in your chest. You're going to force that pressure upwards. Now, when the pressure goes up in your chest, what's that going to do? That's going to push on blood vessels in your chest. The aorta, for instance, a major blood vessel in your chest, and it's going to end up pushing the blood out of the aorta, and one of the places that it will end up going is up to the brain. So when you sneeze and cough, pressure inside the chest goes up, and that pushes blood up to the brain. So the Monroe-Kelly doctrine comes in here. If we increase the blood in the brain, even if it's just temporarily, that's now going to increase intracranial pressure, and when intracranial pressure goes up further, that's going to activate more meningeal nociceptors and therefore result in a worse headache. So that's the reason that the headache is made worse when you sneeze or cough, because you are increasing the amount of blood in the cranial cavity and therefore making the intracranial pressure even higher. So, in the next video we will discuss the accompanying symptoms of the headache that you get with intracranial hypertension, and then we'll go on to discuss different causes of intracranial hypertension.